Understanding the full lineage of materials is crucial for tracking experimental history, troubleshooting workflows, and ensuring data integrity. Until now, our material records only provided parent-child relationships in a table format. With the new Material Lineage View, you can now visualize an entire material's history in a single graph, making it easier to trace materials back to their origins. In this tutorial, we'll explore how a newly generated microbial material in a workflow automatically displays its origins, including the DNA materials used for the microbial transformation. To begin, let's add the necessary materials from the DNA assembly report created in Designs. Click Select DNA Assembly Materials and apply the Entity Type filter to include all primers and primary templates. Once selected, click Next, navigate to Distribute By, choose Table Order, and hit Distribute. Finally, click Create Plate Maps to generate the plate layout. Now, proceed through the workflow, ensuring that when registering new plates, you include plenty of volume to maintain sample integrity. Now, we'll use the PCR and Inventory Check tool to ensure all the necessary materials for the PCR reactions are available in the inventory. This tool helps us locate the aliquots containing the required DNA materials for the PCR process. Here, we can see that all the materials are present, and we'll proceed by selecting the Last Modified option to prioritize the most recent materials. The identified plates will serve as our input plates for the Run PCR task. Once we input the required reaction volumes, we can export the PCR work list. The next step is to execute this work list, enabling us to register the new plates with the newly generated materials. In this new plate, we now have our PCR-generated pieces, which will serve as the essential input assembly components for the assembly our final construct. These PCR-generated pieces are now officially registered in the Material Registry of DNA Materials. After selecting the buildable constructs from our DNA assembly report, the tool automatically detects that all the required pieces for the assembly are available. It then guides us through the assembly planning process, organizing the PCR-generated pieces into a new plate and generating a new work list to streamline the next steps. Again, execute the work list in order to generate new DNA materials of the final constructs. Now, we'll select a microbial material to perform the microbial transformation of our assembled constructs. This step will generate a new plate containing new microbial materials, each carrying the successfully assembled constructs. After completing the microbial transformation work list, navigate to the record view of one of the transformed microbial materials. From there, scroll down to the Material Lineage card and click View Graph. This graph provides a complete visual representation of the material's lineage, tracing it back from the transformed microbe to its parent construct and microbial material, through DNA assembly and PCR reactions, and ultimately back to the original primers and primary templates. With this new Material Lineage View, you can now effortlessly track the full history of materials, making it easier than ever to manage and analyze your biological workflows. If you have any questions or need support, don't hesitate to reach out to our team for assistance. Contact us today, and let's keep advancing your workflows together.